Hello. All right, so we have with us here a biology paper for May June 2025, paper 4.3, all right, which is a Cambridge paper. Okay, so let's navigate through this paper. Let's start with question number 1. It says here, number 1A, figure 1.1 is the diagram of the human digestive system. Alright, we can see the diagram over here, and it has various parts labeled A, B, C, D, E, F, G, H, J, K, L, and M. Alright, then table 1.1 shows information about some of the structures shown in figure 1.1. Complete the table 1.1 using figure 1.1. Here we have our table, so we have to complete this table using this diagram given above. Alright, so here we have the name of the structure, we have to state the function and the letter of the said structure. Alright, so the first part has the structure labeled letter G. We have to go and look for letter G. Alright, so letter G is at this part over here. We can actually see it here. Now, what is the name of this part? The name of this part is the anus, so we'll actually state anus as the name. But apart from that, we have to state the function also. What is the function of this part? The function of this part is ejection. Alright, so we'll state the function over there. Then we have a part that produces amylase. You have to state the name and the letter. Alright, so amylase is usually produced in salivary glands. These ones here, the ones labeled M. So we can state salivary glands over here. Say salivary gland. And the letter was M, so we'll put M over here. And then the gallbladder. What is the function of the gallbladder? The function of the gallbladder is to store bile. So we'll say here gallbladder stores bile. Now, what letter represents the gallbladder? We have to look for the letter also. Okay, so it is this part over here, this one. So we'll label it as letter L. We'll label it as letter L. And then transports food to the stomach. Okay, the part that usually transports food to the stomach is usually known as the esophagus. So we'll label here esophagus and we'll find the letter. Alright, the letter for esophagus is this long tube over here, which is labeled B. So we'll write here B. Alright, then describe the role of chemical digestion. Alright, so chemical digestion, this is a kind of digestion that breaks down food into substances that can be absorbed by the blood. Alright, so we'll say that it is to break down small molecules, small insoluble molecules into soluble molecules that can be absorbed by the blood. Alright, so to break down insoluble molecules into soluble molecules that can be absorbed into the blood. All right, yeah, so this is what the chemical digestion does. Then maltose is digested by maltase. State the name of the product of this reaction. Alright, so maltose, when broken down, what we get is glucose. So we will state here glucose as the product of the reaction. Describe where maltose is digested in the small intestine. Alright, so maltose is usually digested in part of the small intestine, alright? But what part exactly? This part is the membrane of the epithelial lining of the small intestine, alright? So you can say the epithelial lining of the small intestine, alright? Okay, so in the epithelial lining, of the small intestine. This part here is usually filled with cells known as epithelial cells. So when we say epithelial lining, when we say epithelial cells, we mean this is where this digestion is taking place. Okay, let's move forward to the next question. The next question says, explain why amylase digests starch in small intestine but does not digest starch in the stomach. Alright, so the conditions within the stomach are way different from the conditions where amylase acts. Amylase acts in the small intestine. Amylase acts in the small intestine, but it also acts in the mouth. So the condition in mouth and small intestine is way different from the condition inside the stomach. Why so? Because stomach has hydrochloric acid, all right? So it has a higher pH in comparison to, it has a low pH in comparison to the pH inside the small intestine, all right? So we will say that the stomach produces hydrochloric acid, all right? Hydrochloric acid. Amylase does not act in hydrochloric acid, all right? So that's one reason it cannot work over there. But also we know that the acid lowers the stomach pH. Okay, the acid lowers the stomach pH, all right? Okay, then the low pH damages the amylase enzyme. All right, you know, pH is one of the factors that affect enzyme activities. All right, so if it is damaging the if the pH is not suitable, it can actually destroy the enzyme. All right, okay, so once damaged, we say that the damaged enzyme cannot bind to the substrate. All right, so the damaged enzyme cannot bind to the substrate, and then if no binding is taking place, it means no enzyme substrate complexes are formed. No enzyme substrate complexes are formed, and no products are formed, and no products are formed. All right, yes, and no products are formed. Hence amylase, no products are formed in the, hence amylase cannot work in the stomach. All right, okay, then in the small intestine where amylase is working, so in the small intestine there is bile that neutralizes the acidity from the stomach. All right, it neutralizes the acidity from the stomach, and then after neutralizing the acidity from the stomach, what does it do? It provides a suitable pH for enzyme amylase to work. All right, so it neutralizes acidity from the pH, providing a suitable pH for amylase to work and give products. Where, as we know, the product is going to be glucose from the digestion of carbohydrates or maltose. All right, so those are the reasons as to why amylase does not work in the stomach, but it works in the small intestine. All right, we move to the next question. It says here, alveoli are gas exchange surfaces in humans. State 3 features of alveoli that make them effective in gas exchange system. Now, as they are part of the gas exchange system, what are the features that can enable them to keep on working? Alveoli are part of the gas exchange system, so of course they possess the features that are found within the gas exchange surfaces. All right, so what are these features that we can find? First of all, they have large surface area. Okay, they have large surface area to increase area for diffusion of gases. All right, apart from having large surface area, they have good blood supply. Okay, good blood supply, so that the blood can carry oxygen away and bring carbon dioxide towards. But also they have very thin walls. Why? or membranes. Why? Because these membranes shorten, thin membranes shorten distance for diffusion. So those are the features of gas exchange surfaces. Okay. Figure 2.1 is a diagram of the human breathing system. As we can see, it has a part labeled cartilage, it has a part labeled rib, it has part labeled P and Q. Alright, P and Q are representing the intercostal muscles. And then it has a part labeled S. Three muscles involved in inspiration are labeled P, Q, and S. Identify the letters in 2.1 that label the muscle that contract during inspiration. Alright, so the process of inspiration involves breathing in. Alright, now what is contracting during inspiration? It is the diaphragm, they are labeled S, but also the external intercostal muscles, the ones that have been labeled P. So here the muscles will be P and S. And then state the function of cartilage in the breathing system. Now we have seen cartilage here on the trachea. Alright, now what is the function of this cartilage? The function of the cartilage is to prevent collapse of the trachea. Alright, so air can keep on flowing. So we'll say here the function is to prevent collapse of the trachea, and bronchus. You can see in the bronchus, you can see that the cartilage is running from the trachea to the bronchus. So it is there to prevent collapse of the airway. Alright, state the balanced equation for aerobic respiration. Now we know that aerobic respiration, this one here, usually takes place in presence of what? Of oxygen. Now how do we write aerobic respiration? The symbol, it's glucose reacting with oxygen. Glucose is written as C6, H12 or 6, but it's like reacting with oxygen. How many molecules of oxygen? 12 molecules in total. We put a 6 here. Then what will it give us? When it breaks down, it's going to give us 6 molecules of carbon dioxide plus 6 molecules of what? Of water. So this is the balanced equation for aerobic respiration. Alright, during physical activity, breathing changes. Explain the mechanism that links the increase in physical activities to the changes in breathing. Alright, so what are the things that are taking place within the body during physical activity which can result to changes in breathing? First of all, it is increase in carbon dioxide concentration. Alright, because as someone is working out, it means more rate of respiration has increased. And if rate of respiration has increased, it means more carbon dioxide is being produced. So the first thing here will say increase in carbon dioxide concentration. Alright, increase in carbon dioxide concentration. That is the first thing. Apart from increase in carbon dioxide concentration, we have said that the rate of respiration is going to do what? To increase. Alright, so if the rate of respiration is increasing, what is resulting into this thing? It is a high rate of muscle movement. Alright, so increase in muscle contractions. Okay, increase in muscle contractions. Alright, yeah, so increase in muscle contractions will also result into changes in rate of respiration. Yeah, but also we know that increase in rate or depth of breathing. As long as someone is exercising, the rate of breathing is not the same. Alright, so if we have an increase, it means this one will result to changes in what? In breathing. So we'll say here increase in breathing depth. Increase in breathing depth. Yeah, these ones here will all result into changes in breathing. Okay, let's move forward. Question 3 says, complete the sentences about the control of blood glucose concentration. Blood glucose concentration increases after a person eats a meal containing carbohydrates. This causes the dash to release insulin. Now, who releases insulin within the body? It's the pancreas. Alright, so this causes the pancreas to release insulin. Insulin causes the dash to remove glucose from the blood and convert it to. Okay, so when insulin is released, it usually stimulates the liver. Alright, so it stimulates the liver to remove or to convert glucose from the blood and convert it into what? Into glycogen. All right, into glycogen, a form in which glucose is stored within the body. The control of blood glucose concentration is an example of something we say negative feedback. All right, it's an example of negative feedback because it depends on the concept of a change stimulating another change. All right, now outline the treatment for type 1 diabetes. All right, one way we can treat type 1 diabetes, first of all, is using insulin injections. All right, so we'll state here that one way is through insulin injections. All right, but apart from insulin injections, this other thing we can do is monitoring the diet that people are taking in. All right, how are we monitoring the diet? It is through monitoring or counting carbohydrate intake, because carbohydrate is the one that is resulting into production of what? Of glucose. All right, yeah, so here we can talk about monitoring carbohydrate intake. Okay, monitoring carbohydrate intake, because carbohydrates are the ones that are producing glucose. All right, so those are the ways in which we can treat type 1 diabetes. All right, so the question says the scientist investigated the effect of injection of glucose solution concentration in a healthy person. The injection was given at 0 minutes. So we can see on the y-axis the concentration and the time taken. All right, calculate the percentage change in blood glucose concentration from 5 minutes after the injection to 30 minutes. All right, so we come here at 5 minutes, the concentration will be 210 according to the scale. So at 5 minutes, it's 210, but what about 30 minutes? At 30 minutes, it's going to be 120, all right? This is 100, 110, 120. So how are we going to get this difference? We'll take the reading at 30 minutes, which is 120, minus reading at 10 minutes, which is 210. And then this whole thing, we divide it to reading at 5 minutes, which was 210, and then multiply it to 100, and work it out in the calculator, all right? When I work, and the answer should be in two significant figures. All right, so this is approximately equal to, all right, negative 43. All right, times 100% is approximately equal to negative 43. So write the answer here, negative 43. All right, adrenaline also affects blood glucose concentration. Suggest how this is an advantage for the fight or flight. All right, so adrenaline usually stimulates breakdown of carbohydrates to release glucose, or it stimulates conversion of glycogen to glucose so as to produce what? To allow glucose to be used in the process of what? Of respiration, all right, to release energy. So we'll say here adrenaline increases glucose concentration to be used in energy for respiration that allows muscle contractions. All right, that allows muscle contraction. Okay, yeah, so the muscles will contract and be used for movement, which is flight or fight. How does adrenaline increase glucose concentration? It increases the concentration by breaking down carbohydrates or allowing conversion or stimulating conversion of glycogen back to glucose. All right, question number 4 says, figure 4.1 is a photography of water lily in a lake. 4.2 is a photography of a cross-section of a part of water lily with parts labeled A, B, and C. Identify parts labeled B and C in this figure. Part B is the palisade mesophyll, and part C over here is the lower epidermis. So we'll say this is the palisade mesophyll cell, and part C is the lower epidermis. Right? Yeah. And then describe two functions in water lily of the part labeled D. Now we need to see part D. Part D is this part here. This is representing the sponge mesophyll. Now what is the function of sponge mesophyll? One, to allow diffusion of gases. All right. It allows diffusion of gases from respiration, but also from the process of photosynthesis. But apart from that, it allows diffusion of water gases to be used for transpiration, right? So again, to allow diffusion of water vapor, to allow the process of transpiration to occur. Okay, a green pigment is found in tissue B in figure 4.2. Describe this function of this green pigment. Part B is the palisade mesophyll. Now, what is the tissue that is found in it? It is the chlorophyll. All right, now what is the function of chlorophyll? First of all, its function is to trap sunlight. All right, after trapping sunlight, what does it do? It converts sunlight energy to chemical energy for the process of photosynthesis. This is what it does. Water lilies have a very thin cuticle compared to a plant growing on land. Explain how this is an adaptation for water lilies. Alright, first of all, they are in water, so they do not need to prevent water loss at all. That's why they have very thin cuticle. Alright, so say here, they do not need to prevent water loss. Alright, that's the first thing. But apart from that, we say that they need to allow light to pass through. So another reason would be to allow light to penetrate. Alright, and of course they use very little material to make the cuticle. Okay. Table 4.1 shows the stomato density and habitat of two different hydrophytes. As we can see, the hydrophytes are pondweed and water lily. The habitats are pondweed is underwater, but water lily has leaves on the surface of water. The stomato density on upper and lower surfaces for pondweed is 0, but the stomato density for water lily on the upper surface is 573 and 0 on the lower surface. Now, convert the stomato density of the upper leaf surface of water lily to stomata per centimeter square. We need to know that we are given the data in millimeter square. Now, in order to convert it to centimeter square, we are going to square the numbers also. 1 centimeter equals to 10 millimeters. Now, if we are changing it to centimeter square, it means it will be 10. All right, 10 times what? 10 squared, 10 times 10, which will be 100. So to convert it, will definitely take the 573 millimeters square and multiply it to 100 centimeters what? Square. When we multiply it, we'll get 57300. All right, so the stomata per centimeter square will be 57300. All right, using the data, explain the stomato density found in the two different hydrophytes. All right, so to explain the stomato density, first we look at pondweed. Pondweed has no stomato density at all. All right, so we'll say that pondweed has no stomata as it is underwater. All right, so if it's underwater, the gases diffuse from water. All right, so they diffuse from water into the leaves. All right, yeah. And then for water lily, we say that water lily has stomata on upper surface, to allow diffusion of gases and water vapor. All right, and no stomata on lower surface, and no stomata on lower surface as the surface is on water. All right, yeah. So this is the distribution of stomata in these two hydrophytes given. Okay. Figure 4.3 is a photograph of rice, a monocotyledonous plant grown in flooded fields in water-lodged soils, right, as we can see it. Identify a characteristic visible in 4.3 that can be used to classify rice as a monocotyledonous plant. Okay, one thing that can be used to identify it, first of all, you can see it has very thin leaves or very narrow leaves. So we can say here it has narrow, or you can use the word thin, what, leaves. These are the features that we can see of that particular monocotyledon. Okay, water-lodged soils contain a very low concentration of oxygen. Suggest how this limits the amount of proteins a plant can make. Now, low concentration of oxygen means low rate of respiration. And if we have low rate of respiration, it means we have low rate of absorption of nitrate ions needed to make amino acids. So less amount of nitrate ions will be absorbed, less amount of amino acids will be made, and of course low amount of proteins will actually be made, and of course will be low amount of what, of protein. So here we say that less oxygen amount or low oxygen amount results into low rate of aerobic respiration and low amount of energy produced. Okay, so less ATP energy causes little amount of nitrate ions to be absorbed by active transport in the roots. Okay, then low amount of nitrate ions absorbed results into little amount of amino acids made, and hence only a little amount of protein is made by the plant. Yeah. Okay, we move to the next question. Question number 5, which says penicillin is an antibiotic. State the type of organisms that antibiotics are used to kill. So antibiotics kill bacteria. All right. Okay, it is important to try and limit the development of antibiotic resistance. Describe ways of limiting development of antibiotic resistance. One way is completing the dose or the course. This is one way. You only take the dose completely so that you do not make them resistant. But also using the antibiotics when necessary. Okay, so using the antibiotics when necessary, not every now and then. No, it's only when prescribed by the physician. Okay, penicillin is produced by an organism called Penicillium chrysogenum. Figure 5.3 is a diagram of Penicillium chrysogenum. As we can see, it has spores, has cell wall mode of chain, it has hypha. State what penicillin in the scientific name Penicillium chrysogenum represents in the binomial system. Alright, in the binomial system, we assign two names to an organism. The scientific name usually has the genus name and the species name. The genus name, it comes first and starts with a capital letter. Now, as we can see it over there. So, this one here, Penicillium, represents the genus. Using the information in figure 5.1, identify the kingdom that Penicillium chrysogenum belongs to. Okay, the genus, as we can see here, the cell wall is made of chitin. Now, organisms whose cell walls are made of chitin belong to fungus. So, the genus will be fungus. Alright, the kingdom would be fungus. Penicillium can be made in a fermenter. State the name of one other product made in a fermenter. Okay, apart from penicillium, we have hormones like insulin, we have nutrients like amino acids, alright. So other products made here, we can say amino acids, alright. We have the diagram here. Figure 5.2 shows a fermenter where we can see penicillium, amino acids, and carbohydrates are being entered into the fermenter. We have this part which contains the water jacket. We have thermometer, we have gas X entering, we have the stirrer, and we have this part here which is used to absorb the product. Okay, gas X is in figure 5.2 is needed for production of penicillin. State the name of gas X. This gas here is oxygen gas. Alright, so we'll state the name of the gas here as oxygen. Alright, the concentration of gas X is one of the conditions that must be controlled to ensure penicillin is produced. State other conditions required to produce penicillin and describe how they are controlled in the fermenter. Okay, another things or conditions, first of all, is sealing the fermenter to prevent contamination. So here we'll say sealing to prevent contamination. Alright, this is another thing that is done. Apart from sealing to prevent contamination, we have a stirrer over here to ensure uniform distribution. Alright, so we'll say here stirrer to ensure uniform distribution of substances that are within the fermenter. Apart from that, we have a thermometer to monitor temperature. We can see over there. We have the thermometer to monitor temperature. All right. Okay, so here, thermometer to monitor temperature for enzyme activity. All right. Yeah, because we know that temperature can affect enzyme activity. So we need a thermometer to monitor temperature, but also monitor pH. Monitor pH for enzyme activity, because pH also can damage the enzyme activity. All right. Okay, but apart from that, we have a part that usually removes what? Waste products, so as to ensure that they do not damage the product. So removal of waste gases like carbon dioxide and all other stuff, so that there's no competition, yeah, and stuff like that. Right, but also we have the water jacket to regulate what? Temperature over here. Pap, right? Yeah, so we have water jacket to regulate temperature. We'll state here water jacket to regulate temperature. All right. Yeah, so these are other parts of the fermenter that are necessary so as to ensure that it is working to give us the desired product. State the name of the organism that is used in bread making and in biofuel production. All right, so in bread making and biofuel production, the organism that we need is yeast. Okay, move to the next question. It says here, in figure 6.1, the diagrams of an island show the land covered by forest in 1982 and 2013. When you look at the diagram in 1982, a very large portion is covered by forest. In comparison to 2013, very little land is covered by forest. Alright, describe the change in land covered by forest between 1982 and 2013. So, the description is that less land is covered by forests, okay, in 2013. So, less land is covered by forests in 2013. Compared to 1982, all right? Yeah. So if that is the case, we can say that more land more forest has been cleared, so the land is just bare, all right? So if that is the case, it means we have fragmented forests or a broken forest, okay? Yeah. All right, so we'll say here, less land is covered by forest in 2013 compared to 1982. Most of the forests is fragmented. It's just some pieces, all right? It has been broken. Okay. State 2 possible causes for change in land covered by forest. Okay, what could be the causes that have resulted into this? One could be agriculture. People cleared the land so that they could do what? Cultivation of different crops. So agriculture, all right? But apart from that, we could have climatic changes. Alright, so climatic changes, maybe now it's way hotter than before, so most of forests are dying out of high temperatures. Okay, B says figure 6.2 is a diagram of the carbon cycle. As we can see here, carbon dioxide in the atmosphere, we have arrows over there, plants, animals, decomposers, V, and fossil fuels. Alright, state the name of the processes labeled T, U, and V. Now let's start with T. We have carbon dioxide in air. All right, apart from having carbon dioxide in air, then we have process T, then we have plants. Now, carbon dioxide in air, how is it used by plants? It is used in the process of photosynthesis. So we'll say here, photosynthesis. And then we have U. U is like animals, U, carbon dioxide in the air. So animals producing carbon dioxide into the air. This is respiration. And then we have V. Now V, it's like fossil fuels bringing carbon dioxide back into the air. This is combustion. Burning of fossil fuels is what results into increase of carbon dioxide in the air. Deforestation is linked to an increase in global temperatures. Explain how deforestation can result into increase in global temperatures. All right. First of all, deforestation results into a less number of plants. So we have decreased rate of what? Of photosynthesis, which results into increased carbon dioxide concentration. So here we'll say deforestation decreases number of trees, resulting to decrease in photosynthesis and increase in carbon dioxide concentration. All right, an increase in carbon dioxide concentration, which is a greenhouse gas, that enhances global warming. All right, so deforestation results into decrease in photosynthesis, which results into increase of carbon dioxide concentration, and as a result, it allows for accumulation of this greenhouse gas, which can result into global warming. Okay, describe other undesirable effects of deforestation. Okay, what are the other undesirable effects? First of all, if we are cutting trees, it means we are allowing some species to go extinct. So some other species can go extinct because we have destroyed where they used to live. All right, now they will disappear. All right, so extinction of species, but also loss of habitats. All right, because plants or other species cannot survive anymore. But apart from that, we have disruption of the food web. Okay, so species will go extinct due to lack of habitat, due to lack of what? Places to live. So here we can say disruption of food web systems. Disruption of food webs, so plants don't get where, or other organisms don't get where to eat anymore. Apart from that, we can have desertification. All right, so the area becomes very bare. We have minimum amount of energy that is being produced. All right, yeah. Okay, yeah, and that was the last question for this paper. Again, thank you.